This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 933, recorded on September 2nd, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, everybody. Out my window today, it looks good, and it is good. There's a light breeze. The temperature is in the low 80s. The humidity is probably about, I would guess, 40 to 45%. Just sticking my hand out the window, I might be wrong. But it's comfortable. It's a lovely day.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 84 degrees Fahrenheit, which is 29 Celsius, and it's also a very nice day.
And from Madison, New Jersey, Brianne Barker.
Hi, everyone. The beautiful days continue here in Madison as well. 78 Fahrenheit, partly cloudy, but mostly all I see is sun.
I was at Penn Station the other day, and one of the train lines was up, and it was like, blah, 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 Madison, the fourth stop on the train. Right? Very convenient.
That's it.
What is that, about 30 minutes, 35 minutes?
On
40?
the express train, which would be the one you're talking about, it's probably about 30 to 40.
Okay, that's an express. Cool. All right. Well, we have a few announcements for you before we dive into science. And the first is an advertisement for a research assistant in Amy Rosenfeld's laboratory at FDA. And their focus of research is to elucidate the mechanisms by which cross-species anti-antivirus antibodies may affect viral pathogenesis. So this is for a research assistant to support work on picornaviruses like Entero68 poliovirus and human rhinoviruses, includes genetic, biochemical, cell-based studies for understanding multiple aspects of immune dysfunction associated with infection. The person will carry out experiments in cell culture, participate in development of small animal models for studying multiple aspects of virus infection, including immune responses, and all work will be done under BSL-2 conditions. You'll need a BS. You will need to have educational emphasis or strong background in molecular biology or cell biology. Immunology is preferred. Cell biology and immunology is preferred. Experience with mammalian cell culture, virology desired, and previous experience working in a research lab studying immunology, molecular cell biology, microbiology, or biochemistry. Courses equivalent to a BS in science plus appropriate experience or additional education is acceptable. And I will put a link in the show notes. I'll put this whole PDF in the show notes for you, and you will find there Amy's FDA email at which you can email her for more information. A couple of people have been asking for transcripts, and so I thought I would tell you where you can get those. For Daniel's clinical updates, there are transcripts made. They're over at his website, parasiteswithoutborders.com/clinical-updates. And you'll see a big box for each episode. And if you click on it, there you go. Transcripts are there. He has somebody who does those, I think. And then for TWiV like this, we don't do transcript, but YouTube does, okay? So what you do is you go to youtube.com/profvrr. You pick the video you want. And I think it's your selection to have closed captions enabled. Underneath the title of the video, there's a 3-dot menu icon. You click it and you say show transcript. And a separate box will open and you'll see the transcript, everything everyone says with timestamps. And it's not perfect and it doesn't have our names. But it's a transcript. You could actually copy the whole thing if you'd like and paste it into a Word document or your text editor of choice and search for it. And so people always ask for this. Most recently, someone said, I want to find out when you really said the vaccines wouldn't be sterilized. So she wants to go through all that. So I said, sure, go for it. So that's how you can do it. Anyway, I'll put this information in the show notes as well. And Kathy has an announcement.
Yes, another public service announcement. This one is about the BioInc Prize, and we've talked about this in the past. They award this prize every 2 years, the MW BioInc Virology Prize, for an internationally renowned researcher who has made a groundbreaking contribution, or maybe groundbreaking contributions, plural, I'm not sure, but they say one, to virology research in the broadest sense of the word. The laureate receives a monetary award of 35,000 euros and a medal bearing the likeness of MW BioInc. And you can find who previous laureates have been and all the information you need at a website, which we'll put the link in the show notes, but it's KNAW-webpage. And who can submit a nomination: individual scientists, universities, research institutes, scientific organizations, and institutions. And the deadline is October 30th, 2022.
Cool. Ralph Bartenschlager was the last one, and Eva Harris before him. Cool. Very good. All right. Onwards, folks, to the literature, as Mark Crislip used to say. He used to say that on his podcast, Puscast, Infectious Disease Puscast. And now on to the literature. And Daniel and Sarah have taken that in their rebirth of Puscast. We have 2 papers today. I'm no longer calling them a snippet in a paper in response to pressure. 2 weeks ago, I was pressured on the live stream to get a shingles vaccine. So I did. I got one on Friday night.
Nice.
And just a little soreness in my arm. A lot of people say, it knocked me out, which I believe, but I had no issues. So even the pharmacist who gave it to me said, it's a good thing you came at 8 p.m. on a Friday, so you don't have to do anything. It's okay. Pressure. Nothing like pressure.
Yeah, I had virtually no reaction to mine either.
Because we're born in the same year.
Yeah.
Okay, so our first paper is a Journal of Virology paper. Discovering genomic characterization of a novel henipavirus, Angavochli from fruit bats in Madagascar. Dixon, have you been to Madagascar?
Not yet. Nope. I was going to go.
Brianne, have you?
I actually met someone from Madagascar, and she invited me to join her. She studies lemurs. And I said, I would love to go and visit you sometime. And the next 6 months, they had an enormous plague outbreak.
Wow.
And it trashed the entire island, basically, and cut off all kinds of communications and everything. Then they went into a severe drought. I said, maybe I'm not... Those poor lemurs, I felt sorry for them.
I remember the plague outbreak was around the time I last went to South Africa, because there were a lot of signs and sort of travel issues about going between South Africa and Madagascar at that time.
Yep. And, I think it's endemic, and I think it's still going to be there. Smolders, and then every now and then it surfaces.
Kathy, have you been?
I have not.
Neither have I. Could be interesting. I think David Quammen has been. He's been everywhere to visit the lemurs. Lemurs are interesting because they are an ancestral SIV integrated into their genome, right? Which is really interesting. And so at some point they must have come from the mainland. They rode on a branch or something. All right, anyway, back to Madagascar and fruit bats. This is a paper from UCSF, University of Chicago, some institutions in Madagascar, Cornell University, the Institut Pasteur in Dakar. And the first author is Charlene Madeira. The last author is Cara Brook. And there's no shared first authors, right? I didn't miss it. And I thought this would be useful because we could review Henipaviruses. And TWiV 618, which I did in Singapore in December 2019, was a discussion of a human Nipah vaccine, which is interesting. So the two big pathogenic human viruses, Nipah virus and Hendra virus causes outbreaks in racehorses in Australia. And so they developed a Hendra glycoprotein vaccine for the racehorses because the human cases are too few to vaccinate everyone. And that same vaccine protects humans against Nipah virus. And we talked about that in that TWiV 618. And I think it's been through phase 1 already and possibly is going beyond. But,
Vincent,
Nipah
is
outbreaks.
the vaccine any good for pigs?
I don't think they're
pigs
testing
play
it
a
in
big
pigs.
role in this, right?
They do, but the thing about pigs, Dixon, if you put them inside, you don't have a problem. The problem in Malaysia was they were outside. The piggeries were outdoors and they got contaminated by bats, right? So the solution to that was to bring the pigs indoors. And
they have no indoors.
Well, now the pig farms are indoors now. They're biosecurity.
In Malaysia? Really? I didn't know
Yeah,
that.
they changed it all. Yeah.
I see.
But the horses, of course, are in barns they leave the barns and they go eat in the pastures. And so we learned at the bat meeting, the bats will grab a piece of fruit, Brianne, right? Suck out the juice and throw the pulp on the ground, and the horses come over and eat it, the pulp, so they can get infected. So you can't stop that because the horses do need to get outside. But the pigs don't, sadly. I feel badly for the pigs.
So this cross protection, once it's through all the clinical trials and everything, would be really important because the paper said that there's fatal encephalitis nearly annually in South Asia of Nipah virus.
That's correct.
So
that's correct.
It's much more a problem than the horse to human transmission.
That's right.
And the one other thing that's sort of interesting is that there was another Henipavirus that was described in August, infecting some patients in China. So they had some disease that looked like Langia virus.
Yeah, that's right. So these viruses, Henipaviruses, that's a genus within the family Paramyxoviridae. And the Henipaviruses include Hendra virus, Nipah virus, Cedar virus, CEDAR, which is an interesting one because, so Hendra and Nipah, you need to work at BSL-4 because they're really dangerous for people. But Cedar doesn't cause disease. They find a lot of seropositive people, and so you can work with it under BSL-2 conditions, and many people use it as a model. I learned this at the bat meeting. Ghanaian bat virus, rodent-borne Mojiang virus, shrew-borne Gamak and Dariong viruses, and then Langia Henipavirus, which, as Brian said, just found recently it was found in febrile patients in China, and then they found it in shrews. So Hendra and Nipah, of course, infect people. And some of these others, there's serologic evidence. Mojiang was first identified in an outbreak of pneumonia in Chinese mine workers, all of whom died after infection. Yep. And the Langia was identified in sentinel surveillance of human febrile patients with a history of animal contact, and it was found in shrews. So, they have bat and rodent hosts, and they can spill over into people. So, and we know Hendra and Nipah already spill over, so and cause severe disease. So these are important viruses. We need to know what's out there, right? And that's part of this study. The many of these viruses bind to cell receptors, which are members of the ephrin family of proteins. And I had to think about this a while, and I know Kathy did as well. So
I went to the Wikipedia page.
Yes, the Wikipedia page. So ephrin, so on the cells, there are ephrins on the cell surface. One is a receptor, and the other is the ligand for the receptor. So we have an ephrin receptor and an ephrin ligand. And they're both on the cell surface. They have roles apparently in signaling because they're tyrosine kinases. So the viruses bind to the ephrin ligand, which is, a little counterintuitive, right?
Right. But the ligand for this
ephrin
cell
receptor,
interaction is a receptor for the virus interaction.
So some of these henipaviruses bind ephrin ligand, but a few, we don't know what the receptor is, including this new one that we're going to talk about today. So the genomes of these viruses are negative strand RNA. They can be pretty long, like 18,000 bases. They encode 6 proteins, including your nucleocapsid, glycoprotein fusion, F protein, and an RNA polymerase, also a phosphoprotein. And one of the things I've always liked about these paramyxoviridae is that the genome length follows the rule of 6, which was coined many years ago, where the genome length has to be a multiple of 6. Otherwise, it doesn't work. If you make it one base less, you lose infectivity. Something to do with replication and the way the genome is edited. It's really cool. But what's also cool about these genomes is that they undergo RNA editing. In other words, bases are added that are not templated in the genome at the level of mRNA synthesis. So you can get some different proteins made depending on the particular henipavirus. So there's an editing site in all these genomes except CEDAR, which is quite interesting. And then the polymerase can insert additional bases there. It kind of slips and adds additional bases, and that can change the reading frame of the message, and you get different proteins. And the different proteins, which are called V and W, are involved in immune evasion, right? So that's cool. Maybe CEDAR doesn't evade, and maybe that's why it's attenuated.
So, Vincent, they refer to that in the paper as editing.
Yeah.
But I know editing from another perspective, and that is with trypanosomes and humans as well.
Yeah, that's right.
And that's a change of the base that's already there rather than putting in a new base.
Yeah, it's another kind of editing.
I just thought I would throw that out to let you know that I'm still alive.
Do you think that this editing of insertion or deletion or whatever is a way of dealing with the rule of 6? A way to explore the sequence space by adding nucleotides because you can't do it at the replication level?
Yeah, because this is at the mRNA level, so you don't have a rule of 6, right? Yeah, it could very well be. So yeah, editing can be adding bases, or as Dixon says, changing the one that's there.
Yeah, I think either way, it's just anything that's not templated.
Not templated, yeah. All right. So in Madagascar, Henipa and Nipa are, the reservoirs are fruit bats, in particular Pteropus genus, and these are very large animals, beautiful, absolutely gorgeous animals. And they have them in Madagascar, different genera. They have Pteropus, they have Rosettas, and they also have Eidolon. And you know what's weird? I don't understand. Throughout the paper, they give the genus of 2, but not Eidolon, right? They just call it E. duprianum. And I thought, well, is it really hard to spell? But it's not really.
They're saving space.
Saving space.
But why not save the space for the other?
Yeah, all of them, right?
This genus.
So there are 3. There are 3 Madagascar fruit bat species, and they have these viruses in them, and so that's what this study is all about. And one thing I did not know: these fruit bats are consumed widely in Madagascar as a source of food.
Oh, sure.
Yeah, you think now? You knew that, Dixon?
Well, they'll eat everything, including lemur. So they've got
small.
A terrible problem in that they're trapped on an island.
Yeah,
it's a very large island, but nonetheless. And when the people got there, they started to deforest everything. And when that happened, they started to run out of food. And it sounded like an Easter Island problem magnified times a million or something else. And yeah, I think fruit bats are eaten everywhere.
So obviously when you eat them, then you're going to be contacting them. And if they have viruses, you could get them. So that's the issue, right? So you need to know what's there. You could also
imagine it's not just when you're eating it, but also when you're cleaning it.
Yeah, cleaning it. Yeah, that's right. That's right. Yeah, there you have all the eating. It's cooked, right? Presumably.
Yeah, exactly.
I hope so.
Yes, the cleaning, the capture, and the you try and capture it, and it scratches you, right? What did...
I'm also checking to see how big they are because you said, they're so small. So I wanted to know
I meant the
size.
lemurs.
Oh, okay.
Well,
I was wondering about
big.
this Eidolon bat.
So I was talking to Raina Plowright at the bat meeting. I said, do you ever try and capture one of these flying foxes, the big fruit bats? And she said, oh, yes, it's very interesting. She said, you think you've got their legs, and then their little finger on the edge of their wing comes around and tries to poke your eye out.
Oh, no.
The wing has that sharp finger at the end.
Yeah.
Well, what do I know? I've never captured a bat.
Well, according to the internet, always a reliable source, Eidolon can grow up to 202.5 millimeters.
Wow.
Okay.
So I guess that would be kind of small.
So Brianne, this you would get. So it says in the Wikipedia page of Eidolon, the Madagascar fruit bat, Eidolon dupreanum, mainly consumes fruit, but also feeds on the flowers of eucalyptus trees.
Ooh, that's
Remember?
important.
So Raina was telling us,
Yes.
in Australia, they have a problem is that they're destroying the feeding grounds of the fruit bat. So they migrate somewhere else. And she said, all we need to do is plant eucalyptus trees and they will stay there. And now there you go.
That will stop Hendra outbreaks.
Yeah, that's for Hendra, right. So in this study, they collected urine from 206 bats in 8 roosting sites across the island from 2013 to 2019. They collected them during the wet and dry seasons from all of these 3 bat species, P. rufus, E. dupreanum, and R. madagascariensis. They isolated RNA from the urine. They did deep sequencing. And they looked for henipavirus sequences. 4.9% of the bats were positive. 10 out of 206 were positive for some kind of henipavirus. And all the positives were from E. dupreanum. All 10. Yep. And they got these positives at the Angavokli cave roosting site. Hence, they call the virus after the cave. And they got positive samples both in wet and dry seasons from both male and female adults. And I think that's interesting because they have different feeding behaviors in the different seasons. You want to know if there's some prevalence of virus during one or the other. They got one near full-length genome, 16,740 bases. Not complete, but almost full, and they focused the whole paper on that. And so they call it Angavokli virus. So it's interesting. They name it after a place, right?
After a cave.
Yeah. I guess nobody minds if... But
they have not yet found a disease association.
Yeah, maybe that's it. Because Muerto Canyon was a little canyon where Sin Nombre virus was first found, and the locals didn't like them using that name.
Yeah, but maybe people lived in that canyon. Or
it could be.
Nobody lives in the cave.
All right, but we have Angavokli virus. The genome is typical paramyxovirus, as I told you. Nucleotide identity, 36% average nucleotide identity with Nipah virus reference genome, so quite distant. And you can see all the proteins, the P gene, the editing site, which would give the V and the W proteins. The glycoprotein, a little bit longer than previously, 56 amino acids longer. They can see nucleocapsid, matrix, polymerase proteins. And by the way, the polymerase has the highest nucleotide and amino acid identity with the others. So it's very conserved protein. You could theoretically design antivirals against that. There are also non-coding regions between the coding regions, which is typical. What's interesting is the phylogenetic analysis of this virus makes it basically ancestral to all currently known bat-borne henipaviruses. And they estimate divergence of this lineage from the rest of the henipaviruses of bats 9,794 years ago. It's very precise. But the range is 6,500 to 14,000 years. The if you're interested, the time to most common ancestor for the entire Henipavirus genus is 11,195 years ago. So the virus that gave rise to all the Henipaviruses we estimate was circulating 11,000 years ago. So this is an ancestral virus to what we know. Of course, as we isolate more, that we'll be filling in this tree.
But it also tells us that these viruses have been around for a while.
Longer than us, right?
Years. Yeah.
Longer than us. So then they ask, could this glycoprotein bind ephrin? There's some residues that we know are on the other Henipavirus glycoproteins that interact with ephrin. None of those are here, so they think it's probably not binding to ephrin ligand. So that's it. I thought it's cool that now we have one of these in Madagascar. A few closing remarks. They say in fruit bat systems elsewhere, Henipaviruses are shed in urine at higher rates during the nutrient-poor dry season.
Right.
And in the case of Nipah and Hendra, these seasonal viral shedding pulses have been linked to zoonotic spillover.
So do you think it's because it's nutrient poor that they're more prone to be infected and therefore they are going to shed more?
My understanding from the bat conference was that during those nutrient poor times, they were under more stress and were shedding more.
Yeah. Because I think they believe the population is infected all the time and they have these pulses, right?
Yeah, and this was also the stuff
which
that
can
correspond
to nutrient deprivation or to birthing, right? I think that was another one correlating with it.
Or any other kind of disturbance. And so that's why when you start to think about, land use and deforestation and things like that, you can imagine that contributing to outbreaks.
They talk a bit about viruses in bats, and they make a statement which is very interesting. They say, and this is about beta coronaviruses in Madagascar. So these have also been recently found in Madagascar fruit bats. And these, they say, have surprising identity to lineages circulating in West Africa. And they say this suggests that even these fruit bats, these Malagasy fruit bats, likely experience some form of contact with the African continent. They have 49 bat species in Madagascar, and 9 of those are distributed across Africa, Asia, or Europe. And they say this presents opportunities for interspecies viral transmission via island hopping. Isn't that a thing that tourists do?
It is.
Island hopping. So bats can do it too. Anyway, this is obviously a drop in the bucket, right? You have a lot of authors who have worked a long time on this one virus, but there are many, many more out there. And just look at all the work just to find one. And I wonder if any of the sequences, they don't mention that, right? How different they were, because they only focus on this one. I wonder if the others are from different enough viruses that they would merit independent study.
So the distance from Madagascar to the continent of Africa is 225 miles or 360 kilometers at the closest point. So is that within the range of a single bat?
I think it would be. My understanding was that bats have a particularly large range.
They do.
Because I could try to figure out their specific range, but I did think it was large.
I know the one study out of CDC in Uganda, they said they went 90 some kilometers in one night, right, to get fruit.
Yes.
So I guess that's a lot, but I don't think it's out of the range of some bat species. Yeah.
I Googled what is the range of bats, and I got a bunch of answers about what their echolocation, what frequency they.
Oh, really?
I think it's
not
different.
Their distance range.
I thought you'd get something on baseball bats, but you didn't.
It's all in kilohertz, 9 kilohertz to 200 kilohertz.
There was a fabulous nova on about 3 weeks ago on the agave bat, or the tequila bat, as they call it, down in Mexico. It's essential for the pollination of the agave plant. without the bats, they don't have any tequila. So they were extincting the bats by taking away their habitat. Of course, what else is new? And this guy, who was known as the Batman, according to David Attenborough, sought out and found the largest single undisturbed colony of tequila bats. And what they did was they reintroduced them to the areas that they had been chased out of. And they reinvigorated the tequila industry, which is near Guadalupe, by the way.
And then they had some tequila to celebrate?
Well, let's hope they did.
This just shows that if you give people an incentive, they will protect wildlife.
Yes. And the other thing, these are tiny little bats, too. These are like the size of maybe half the size of a little brown bat. So but there were used to be millions of them, and they were localized to specific caves, and those caves were impacted by agriculture as a result, and then their area was restricted, but they didn't migrate at all. They stayed put no matter what. So some do and some don't migrate, I think.
Okay, I found a bat that traveled more than 2,200 kilometers.
There you go.
From Spain to Latvia.
Oh,
or the other way around, Latvia to Spain, but yeah.
Wow.
But they take the Orient Express to get there.
I think different species can go different distances. This one that, remember we had the guy from Hungary, Pécs, University of Pécs.
Gabor something.
Kameneshi.
Yeah.
Kameneshi. He was at the bat meeting, yeah. Mm-hmm. Those bats have a big range. What was the name? Schreiber's bat, right? That was the name of it.
Yes.
Yeah. Which they are thinking, they carry love you virus, right? The love you virus. Yes. Yeah. Okay. So there you go. And that is paper 1. Paper 2 brings us back to SARS-CoV-2. And I picked this paper because it starts to look at some things in more detail than we've seen before and get some answers. And it's a Cell Report pre-proof entitled Spike Protein Independent Attenuation of SARS-CoV-2 Omicron Variant in Laboratory Mice. And this is from, let's see, we have 2 co-first authors, Shufeng Liu and Prabhuanand Selvaraj. The last author is Tony Wang. And they are at, interestingly, CBER FDA, Amy's division of viral product. Well, not hers, but that's where she is. And also the IBM Center in New York. They did the computational stuff that we'll touch on. The, what is it called? Molecular Dynamics Simulations.
Wow.
Recently, I had a pic of a video from the WeHi. You may remember it, of virus particles. They're all That's a molecular.
Yes,
that is. And one thing you didn't mention with that pic is actually where those simulations are. At WeHi, they're in glass bricks in a wall. So when you walk by the wall, you can watch them.
Oh, they're on display. Oh, cool.
I hadn't. That's a new addition since I was there. That's for sure.
That's cool. So this paper largely explores Omicron and its properties in mice. And remember, Omicron has over 30 amino acid changes in the spike protein, but also some elsewhere in the genome. And this paper wants to ask if the other ones are also important or not. So just to remind you, 30 plus amino acid changes in the spike. Many of them in the receptor binding domain, which can alter the species tropism. In particular, N501Y, which appeared in other variants, allows the virus to bind ACE2 in the mouse. So the original ancestral SARS-CoV-2 doesn't bind murine ACE2. But naturally occurring variants, including Omicron, have arisen that can bind mouse ACE2. And also, if you remember, Ralph Barrick's lab, they passaged the virus in mice a long time ago, and they came up with some of these same amino acids that allow expanded receptor recognition. So and they talk about this. So Omicron can now infect mice, but doesn't cause much disease, right? So that's what they want to try and address, at least in mice. We know that Omicron causes disease in humans, but we also know that mice are not humans, so that's not surprising. So what's going on here? That's the question. And they have a variety of approaches to do this, which I think are beautifully summarized in the graphical abstract, right? It's one of those graphical abstracts, which they have the viruses they made, the mice, and the outcome after infection. So if you take a look at this, you can look at that. So first thing they do, they make pseudotype viruses. They take another virus, in this case a lentivirus, and put the spike of SARS-CoV-2 in it, and those viruses have a fluorescent protein encoded in their genome so that you can use that as a measure of reproduction. They then infect.
They called their negative control the bald virus because it had no
isn't
spike.
that funny? The bald virus. I'm a little insulted. You should be too, Dixon. You don't mind. I'm not insulted at all. I save that for other things.
Yeah.
The bald virus has no spike protein, which is their control. And they use these to infect 293 T cells, where those have been given ACE2 DNA by introduction of plasmids. And they use ACE2 from 10 different species, which is cool. And so most of these ACE2 were allowed infection by either the ancestral spike-bearing pseudoviruses or Omicron spike-bearing pseudoviruses. So with the human ACE2. Mouse ACE2, when they put mouse ACE2 into these 293 cells, they got what they call massive infection by pseudoviruses bearing Omicron, but not the ancestral SARS-CoV-2 spike. And we've known that before, right? So the ancestral SARS-CoV-2 spike doesn't bind mouse ACE2. Interestingly, and we knew this too, horseshoe bat ACE2 only mediates entry of the ancestral. Let me call it WA1, Washington 1. This is the first isolate from the U.S., right? Yeah, that's interesting. So that's very close to the beginning of the outbreak. So it's still interacting with a bat ACE2, but not Omicron. In fact, as the virus drifted away from WA1, it lost the ability to bind horseshoe bat ACE2. Okay, so right there you see this is just a spike. Clearly, the Omicron spike is really good at binding. At least it binds mouse ACE2 enough to mediate entry of a pseudotype. Okay. Now they do molecular dynamics simulations. These are done at IBM, which is upstate New York. So they say we want to understand the spike. In fact, the receptor binding domains ACE2 interaction. So they perform what's called all-atom molecular dynamics simulations. And so these are molecular dynamics are to understand how atoms and molecules are moving, right? And give you a dynamic view of the interaction. It's way more complicated than that, but that's what you need to know. And so they can look at the different RBDs and the different ACE2s. And one of the conclusions that they reach is when now they have a new abbreviation here. Ancestral. What is WH RBD? WH. Wuhan-like, right? Okay, Wuhan-like, which is the ancestral. Okay, when a Wuhan-like RBD engages mouse ACE2, so there's the spike and the receptor. So there's a binding interface. They say there's two negatively charged residues in the receptor that are going to be buried. And that's energetically unfavorable. They can tell this from their simulations, right?
And remember that mouse ACE2 did not particularly allow for entry of
Right.
the Wuhan-like virus. So this is now the structural reason potentially why.
Right. And then they add the N501Y change, which we know strengthens the interaction. And in a mouse ACE2, you now have different charges on the surfaces, which they say electrostatically attract each other. And they say this now features 2 salt bridges at the interface with 0 net charge, which is energetically favorable. So just this one amino acid change is now making an energetically favorable interaction at the RBD spike interface. And they say, a recent study has actually shown that measuring affinity, the Omicron RBD binds mouse ACE2 with very high affinity, as they suggest here. They also see that Y501 has some other changes which are making it a better interactor. So these simulations, they say, these imply that a buried charge inside the protein-protein interface, the RBD spike interface, reduces the binding affinity. But if you have a salt bridge in there, it enhances the binding. So that's in a simulation. So they say, let's test it, which is great. I like that. So they make constructs of an ancestral WA1 spike with a variety of amino acid changes at the 2 sites that are important for enhancing binding, 501 and also 493, 496. A whole variety of different amino acid changes, which they say, based on their simulations, are supposed to strengthen the interaction by making salt bridges. Okay. They also have a spike, an ancestral spike with 3 amino acid changes that were found in the mouse-adapted virus that we talked about briefly. It's their positive control for good binding. They make pseudoviruses with these spike proteins. And they say all of these amino acid changes that they introduced, including the spike with the triple mouse adapting changes, all of them infect 293s with murine ACE2, 2 orders of magnitude better than the ancestral spike. So introducing these changes really improves infection.
Yeah, so their molecular dynamic simulations are sort of borne out nicely by that.
Yes.
And just to remind people that 293s are human cells.
They're human cells, which don't have ACE2, right? So you add it in by DNA plasmids. The other thing they say here is notably, so whenever an author writes notably or interestingly, and they don't tell you why they're saying that, you have to remember that, because at some point, hopefully, it will come back up. So they notably, the Omicron spike and the WA1 spike with these 2 amino acid changes that improve binding, right, to make it more Omicron-like, were as fusogenic as the ancestral spike, as fusogenic. So that's just a result, which is right, because this is a results section. They're not supposed to start interpreting here, right? We do that in the discussion, so we'll get to that later. Okay.
Oh, I just found a typo in there.
Yeah, there's a typo, yeah. Capital the T and the, right?
No, I was actually talking about blab sea mice.
Oh, I found that one too. Yes.
Just another base substitution, that's all.
Wow, that's funny. That's actually a funny type. Well, this is a pre-proof, right? Hopefully they're going to correct it, right?
Very talkative mice.
Okay. So now they want to put these changes in actual SARS-CoV-2 virus, because eventually they're going to put them in mice, which is in vivo. So they put the 2 amino acid changes in the RBD that improve binding to mouse ACE2. They put those in the WA1 genome. So now we have WA1 with 2, including N501Y, right? These efficiently infect mice now because they have these 2 amino acid changes that allow good binding to mouse ACE2. They also cause weight loss. The viral loads, this is all by PCR. If you compare the ancestral virus with this virus with 2 amino acid changes, you have 2 to 3 logs higher viral loads in infected mice. But the mice don't get very sick, right? They have mild disease manifestations like ruffled fur, lethargy, and eye closure. When I get tired, I close my eyes as well. Yes. So the virus is reproducing now because of this change in the spike, although it's not causing much disease. Okay. Next, they infect blab sea mice, right, Brianna?
Yes.
Blab sea mice with Omicron itself, right? Not WA1, but now we have Omicron. And at the point where they're doing these experiments, it's already been published that if you infect either Syrian hamsters or K18 transgenic mice, that is transgenic mice that produce a human ACE2, Omicron is attenuated. We actually covered one of those papers, I think the Syrian hamster paper here on TWiV. So they made another virus which is ancestral with the Omicron spike. So they take out the ancestral spike and replace it with Omicron.
Yeah, and one thing to, for people to remember here is that previously when they were doing the pseudotype experiments, they were able to look at those changes that are maybe related to Omicron in spike, but not in some of the other proteins.
Exactly right.
We always say, oh, well, Omicron does X, Y, or Z because of its antibody evasion or changes in spike or changes in spike affinity. And we sometimes neglect the fact that other proteins are changed and might have different functions too.
Yes.
So actually using Omicron instead of just some of the Omicron spike is an important comparison.
And, to do these experiments, you have to have a BSL-3 laboratory, right? And if you want to use mice, you need an animal BSL-3, which is even more involved. So that's why a lot of people just use pseudotypes with spike. You get limited information, but you have to recognize that, right? You have to say, okay, we don't know this. But here they're doing the infectious virus, the authentic virus in mice. So we have ancestral, and then we have ancestral with the Omicron spike. All right. And then they have ancestral with these 2 amino acid changes that allowed it to replicate in non-transgenic mice. That's what they call their positive control. Okay. So their logic here is WA1 Omicron spike should enter cells in the same way that Omicron does because it has the spike, right? If the original Omicron virus doesn't infect cells in the lung because it can't enter those cells, which they say has been implied in several studies. So a number of studies have said, the Omicron spike must be defective at entry because it's not infecting mice. Well, then they should see that in this experiment with ancestral with the Omicron spike. You should get low viral load because the same entry block should exist, right, if you have the Omicron spike there. On the other hand, if the attenuated phenotype of Omicron is due to poor replication or elimination by host responses, then WA1 Omicron spike should reproduce really well, right? So that should because that's got all the other ancestral sequences there except for Omicron spike.
Yeah, so if is the viral load change based on a spike change or a something else change?
I mean, the point is that people took Omicron and stuck it in hamsters and said, it's attenuated and has so many changes in spike. It must be spike, right? But there are not zero changes elsewhere. So you need to do the experiment. So here we go. All right. So do the experiment. They do intranasal inoculation of these mice. These are Balb sea mice, right?
I believe so, yes.
Yes, Balb sea mice first. Intranasal inoculation, the only group that had modest weight loss was the ones that got WA1 with the 2 amino acid changes. But WA1 Omicron and this other mice, they got clinical signs of illness, but they all recovered. The viral loads in the WA1 Omicron and WA1 Q493R N501 infected mice were much higher than from the Omicron infected animals, right? Because these are wild type mice. They don't have a human ACE2. There's some immune cell infiltration in the lung, but they all recover. But again, the Omicron or the two amino acid changes addition to WA1 allows better reproduction, which we knew. Okay. So these mice don't get serious disease, so they turn to the K18 human ACE2 mice. So it's a human ACE2 gene driven by a highly active promoter. So in those mice, the infection is lethal. So if you want to see a difference in lethality, these are the mice to use because the wild type mice don't die. So you can't look at lethal disease, right? So they inoculate now the K18H human ACE2 transgenic with 3 viruses, WA1, WA1 Omicron spike, and Omicron. They monitor them for disease, and then they measure viral loads. WA1 and WA1 Omicron spike infected animals, severe weight loss starting on day 5. All the WA1 Omicron die. Only one in WA1 infected animals survive. So the WA1 we know is lethal, right? That's the ancestral virus. It's now hitting human ACE2 in these mice, and that's a lethal infection. But if you put the Omicron spike into WA1, it still kills all the mice, or most of the mice, right? But the mice infected with Omicron itself, minimal weight loss, and only one died. And viral loads from those mice were lower than from the other animals. So this is telling us that the spike is allowing entry and replication in these animals.
But that the differences in this pathology are due to changes elsewhere.
Elsewhere, yeah. So Omicron doesn't do well in these mice. And you take the spike and you put it into WA1 and it's not attenuating it. So it's other than the spike, right? So they say, okay, we want to explore this Omicron spike independent attenuation because it has to do with genes other than this that code the spike. They made 2 other viruses. And this is exactly what you would do. You take now Omicron, you put the WA1 spike into that, right? And they use BA1 in this case. And they have another recombinant where they take WA1 and they substitute sequences from Omicron from the 5' non-coding region through the coding region for NSP12, which is not very far into the genome. I think it's still in that left-hand third of the genome that encodes for the RNA synthesis proteins. Okay? There are mutations in the genome in that region, but not all of the non-spike mutations are there. They infect the mice intranasally, and these are, again, K18 human ACE2 transgenic mice. So you infect them with Omicron with the WA1 spike, or Omicron with that bigger chunk of the genome. WA1 with the bigger piece of the Omicron genome. Very minimal weight loss, and then they recover. But if you infect these mice with WA1, right, they're all dead. Again, we've done that a couple of times. So again, let me repeat it. Omicron WA1S, so Omicron itself with the spike of WA1. We know the WA1 spike mediates entry very well into mice, into ACE2 transgenic mice. But if you put that Omicron spike onto WA1, if you put the WA1 spike onto Omicron, now the mice don't get very sick and the viral loads are low. Viral loads from Omicron WA1 spike infected mice were significantly lower than those from WA1 infected animals. And also WA1 5' UTR NSP12 infected mice had lower viral loads. So there's something outside of spike that is attenuating the replication and disease in mice. It's not the spike.
And it implies that it perhaps is some change in that 5' UTR to NSP12 region.
Yeah, and maybe that and more. We don't, as they say, we have to dissect it out. They have to do more recombinants with different combinations of the changes and see what's going on. Now, why is this important? So clearly Omicron spike efficiently binds to mouse ACE2, but the animals get mild disease. We know this. And they say others have proposed that the spike is not efficiently cleaved and is shifting away from TMPRSS2 expressing cells like pneumocytes in the lung and becoming less pathogenic. And others have published that the Omicron spike is less fusogenic compared to, say, Delta. And that's why they said notably in the results section, because they say, we found the Omicron spike mediates cell-cell fusion as well as the ancestral spike protein. And so I asked Gary Whitaker about this. He said, yeah, these are very difficult experiments to do. It depends on the cells. The cleavage of the spike is very tricky to study. And he said he doesn't think that there's a trend towards TMPRSS independent cleavage in Omicron. And you can hear that TWiV in a couple of weeks where he talks about that. So they say, we think there are genetic changes outside of the spike that contribute to attenuation. And so the low loads that we see, the low viral loads that we see in infected mice are not because the viruses can't get in, but something beyond entry, a post-entry block, right? So they say we challenge our results challenge the notion that somehow Omicron fails to reach the mouse lung. It's something else. And they could say it could be a replication defect, or it could be a heightened susceptibility to rodent innate immunity. And it's true. It could be either one. And they said we have to look at these 17 changes and find out which of them might be the ones that are doing it. So I like this because it clearly shows you it's not the spike in this case. The spike is doing other things in people, right? It's evading antibodies. It's very important. But in the mouse, the attenuation is due to outside the spike changes. And what, maybe most importantly, what relevance that bears to humans is not clear, right? Because people dying now are getting Omicron infection. So it's not that the virus is also attenuated in people. It may be attenuated specifically in the mouse model. It would be interesting to know the mechanism of attenuation. And then maybe you could look in people and say, is it possible that this is still happening? If you could limit it to, if you could narrow it down to a few amino acid changes, non-spike amino acid changes, and then show, for example, that they vary in humans depending on the isolate, then that would say that these are not important. So,
but it might also say that there is some importance to the biology of those changes outside of spike in humans. We don't necessarily know that they're doing anything, but we should actually pay attention to them because clearly they have some biological relevance, at least in a model system.
But I guess the point I want to make is that Omicron is not mild in people, right?
No, not at all.
But unless you're immune, right? And then it can be mild. So the early findings of, attenuation in hamsters and mice, okay, in those animals it was attenuated, and here possibly we were getting a glimpse of the mechanism, but it doesn't always apply to people, right?
No, you are 100% right about that. The only point that I wanted to make is that sometimes I am frustrated when people forget that there are changes outside of spike.
Also, it's important to understand this mechanism in mice. People may say, well, who cares if it's a mouse-specific thing, right? Well, no, mice teach us things, right? You say, okay, now here's a mechanism of attenuation. Maybe we can look at infections of people and get some clues as to whether it's happening there as well. So I think we need to follow up all of these observations. So that's why I like this paper. Relatively straightforward, but the right experiments. You make the two inverse recombinants. It's exactly what you'd want to do. Okay. All right. Hopefully you learned something, folks. If not, go back and listen again.
I found this paper had what I consider to be a very short introduction, almost to the point of not matching the standard structure. And the discussion was also really short. I kind of wanted more to help tie it together for me.
I found when I reached the end of the introduction, it was abrupt going right into the experiments, right?
In fact, I thought, did I not get all the pages? And then I had to look at the line numbers and everything's there. And because it doesn't have that last paragraph that sort of restates what they did and what the results are in a different way from what's in the abstract.
However, I think the results are good and interesting. And I think they support their conclusions, right? At least for mice. Dixon, can you take the first email, please?
Happy to do so. Andrea writes, I really enjoyed your recent TWiV episodes. If a bat pees in a forest, does anyone care? Thought you might enjoy this article on Rich's University of Florida bat program and their recent efforts to move them to their new home. Thank you and the whole team for all you do. You are the best science podcast out there, and I look forward to listening every week. The ultimate campus move-in problem, rehoming UF's iconic bat colony. That's the reference that she gives for moving a bat colony. You know, it's not a trivial issue. How are they going to teach them how to home in on the new place?
I'm trying to find the actual article.
Yeah, this link is generic and doesn't have the specific issue or
and what kind of bats are they? So is there brown-nosed bats? Or?
Yeah, I'm trying to find out. The University of Florida faces the unique challenge of relocating hundreds of thousands of campus residents, its beloved bat colony.
Well, the kids are going to get bitten by mosquitoes like you wouldn't believe.
Oh, here, I got it.
I've got the link.
You got it, yeah. I do too. It's bat.house.renovation. What kind of bats are they? Let's see. It takes a while to get into the article before we find that.
I just put in the article that I have.
Okay.
They're not mentioning the bat types just yet. Brazilian free-tailed bats.
Okay.
Tadarida brasiliensis. That's the vast majority of them. That's one of the most common bat species in North America and most common bats in Florida. Make their homes under bridges and Spanish tile roofs, in attics and concrete block walls or expansion joints.
Oh, so they have 3 bat houses and they house 500,000 bats. And crowds gather regularly to watch them streaming their insect-eating bats. Now they're dilapidated, so they built a new bat barn, and they have to get them in there. And they said you could just carry them all in, and then they would leave the next day. So it's not that simple. Very cool. All right, Rich, there you go. Check it out. Brianne, can you take the next one?
Sure. Ben writes, this is Ben from Louisiana. Why is a nasal vaccine not more widely available for COVID? Would a nasal vaccine not be more effective at helping prevent the spread of Omicron with the increased IgA antibodies versus a standard muscular injection? The infection prevention protection of Pfizer and Moderna are only about 3 months. How do we limit the spread if these vaccines only provide enough antibodies for 3 months? Would a nasal vaccine Omicron booster not increase infection protection from 3 months to 6 to 12 months because of the more IgA antibodies in the nasopharynx? Why not skip the trials on these? Because we already know they are safe and effective, so we can control the spread of the viruses more, and they are already FDA approved, I guess. We just need to make them nasal vaccines to get a better IgA response. I'm not sure that we know exactly all of the efficacy of these in terms of dosing and ways that we can deliver to the nose and get good immune responses and also good memory responses. I see what you're saying, Ben, but I'm not quite sure that we are there yet and ready to actually be doing these as intranasal vaccines.
Do you think he's suggesting using an mRNA vaccine as a nasal vaccine?
Yeah.
Okay.
I think so, yes.
But it's a different delivery, so you need to test it, right?
Right, exactly.
Right.
So my question for you, Brianne, is When you do a nasal vaccine, do you get memory B cells in the mucosa? Are there local B cell memory cells?
I'm not sure about in the NALT. In other mucosal locations, yes, but I'm not sure how well the NALT has been studied.
Okay. So it may not necessarily provide anything different from, it may depend on serum antibodies getting there once you have a challenge.
Right, or it may not give you kind of particularly long-term protection.
Right.
The only thing I really know about this is we did do some intranasal vaccine testing when I was in the lab where I was an undergrad in rodents. And it was quite challenging. We had to anesthetize the rodents in order to do an intranasal in them. And we had to spend a lot of time worrying about their sneeze reflex. And when they would immediately sneeze everything back out.
Well, these nasal, go ahead.
No.
The nasal vaccines are in testing, so we'll find out, I suppose, at some point.
Yeah. There's a number of companies and labs that are working on various ways of delivering these. And not necessarily mRNA platform, but other types of nasal vaccets. If you just have to Google nasal vaccines for COVID and you'll get a bunch of hits.
Let me take the next one. It's on polio. Philip writes, does one given the OPV shed after all 4 doses, or is there enough gut immunity after the first or second dose to inactivate the virus before it leaves the gut? All right. So, yes, that's why you do 3 doses, because the idea is that each dose of OPV, you get one predominant virus, one serotype replicating. You get immunity to that, and then you have to come back 2 more times for the other 2. So you come in a second time. Now you're not going to have much reproduction of that first serotype, and now one of the other 2 will win. That's the idea.
So all the doses are trivalent?
Yeah. Well, they used to be. Now they're bivalent because they've gotten rid of the type 2, right? But they still give 3 doses, I think.
Is that because it's easier to not have to track which dose to give you instead of giving you like monovalent 1 and then monovalent 2 and then monovalent 3?
So they used to, when the OPV was first introduced, it was 3 monovalent doses, okay? Which that's what you would do without any other data, right? Just,
right.
But it turned out that
what we got it early on, and when okay.
In fact, I think my card that I have says type 1, type 2, and then type 3, the 3 doses. But after a while, it was found that you were getting a lot of vaccine-associated polio with that approach. So combining them reduced that, because apparently there's some interference. So that's the reason we made a trivalent, in my understanding. Okay. All right. So if the first dose was IPV, would that eliminate the small risk of the OPV causing polio right off the bat? Yes, actually. So that's if you, and this is one of the approaches suggested by WHO. If you're concerned about vaccine-associated polio, give 1 dose of IPV followed by the courses of OPV. You can do that. And in fact, when the U.S. transitioned from OPV in 2000 to IPV, we didn't go cold turkey. We went IPV, OPV first, and then all IPV. But yes, so that would work. And then Philip says, could an oral antiviral given X days after OPV eliminate shedding? It could, but we don't have any. Okay. We don't have any such antivirals. But
Philip is thinking. It's good.
That's a good point. And in fact, the Gates Foundation is supporting the development of such antivirals to try and eliminate shedding in people who shed for years. Right, there are some people without B-cell immunity that get the vaccine and they shed for years. And if you could identify them, you can give them an antiviral and they would stop. So that's a good goal, and it's being worked on. Continuing with Philip, both OPV and IPV are great as polio is rare. Both offer protection but not eradication. It is sad that we are still talking about polio in 2022. I agree. And I think the reason I used to be a big proponent of eradication, I thought it would work, but we didn't realize how well Sabin 2, vaccine-derived Sabin 2 circulates among people. In fact, some of the literature you can read, they'll say its circulation completely outstripped all of our models, right? Which tells you about models. Anyway,
get a new model.
I no longer think we can eradicate because we got these vaccine strains circulating. The only hope is that these new OPVs have time-limited circulation, but we don't know. It is sad that we're still talking about polio. Absolutely. Anyone interested in virology should visit Warm Springs. Now, that would be a great listener pick for ending TWiV. It is fortunate we still have experts like you that understand polio. Dr. Rosenfeld is there to carry the torch as your career winds down. All right, my lab is winding down, but I'm still here. I'm still kicking.
Let's get this straight.
But thank you. Thank you very much. No, Amy took all the viruses and cells, and she's going to do all that. There, you heard the ad there. Yeah, it's great. I will just be on the sidelines doing color commentary.
Right.
Which is, I love.
Talking to the media.
Talking to the media.
You could do the play-by-play.
Kathy, can you take the next one?
Blog Design writes, Dear TWiV, have you heard about Dr. Fauci planning to step down from the NIH? And gives a link. Yes, I get it. He has been in the spotlight for the past 2 years because of politicians spreading anti-vax conspiracy theories in relation to COVID-19 pandemic. At the same time, I go back at some of the TWiV archives back when only medical researchers had heard of him in relation to how medical research funding operates in the USA.
The real question is who's going to replace him?
That is the real question.
I have a prediction.
Who did he replace when he took his job?
Oh, it's a great question. Can we Google it?
I think there are some wonderful people waiting in the wings to
here we go. Previous
assume
directors.
that role.
Richard Krauss.
Richard Krauss, I knew him. He was from Rockefeller. He went down from Rockefeller.
And he was there for 9 years.
Yeah.
Unlike Dr. Fauci, who's been there since 1984.
This is all true. Fauci has turned down more opportunities to become the director of NIH than almost anyone alive. He refused to get involved because there's too much politics. Look what he ran into. There's politics. If you turn over every rock, you can find a snake.
Dixon, can you take the next one?
Happy to do it. Tom writes, in case you missed it, here's an interesting comment on whether data is plural or singular in the English language. And I confess I haven't clicked on that to see which one it is.
If you do, you have to subscribe, so it doesn't matter.
You see that I'm off the hook. My guess is that it's both, but that's another thing. From a devoted TWiV fan who discovered you during the pandemic, please keep up the good work. My motto to my friends is, show me the data.
Yeah.
That should be everybody's motto.
This is, it's in The Economist, which requires a subscription. So you get the first 2 paragraphs when they start to discuss this what do they call it? One of the most curiously polarizing issues ever generated in English. I don't know if it's that polarizing, is it? Anyway, it's fun. Thank you, Tom. Brianne, can you take the next one?
Simon writes, at the moment, there's a lot of discussion as to the limitations of vaccines giving immunity, but not in the site of initial infection. This is true for both COVID vaccinations and polio IPV vaccinations. But here's the thing. In both cases, the vaccination protects you from disease, but not abortive so-called breakthrough infections. However, when such infections occur, this should then result in immunity in the place of infection, aka in the nose for COVID and the gut for polio. The next time you have a better localized response. As I recall, a recent study actually showed this for Omicron COVID infections. So I think that there's also a time piece to all of this. But it is certainly true that if you have a abortive infection, I'll use Simon's terms because I don't like the other word, you would get a nice boost in the site at which that infection happens. So that would certainly help.
Yep. Simon writes something interesting. So I was born in 1964. I had a smallpox vaccination. 1965, my first birthday, didn't scar. The only scar I have, I believe, from my BCG. By the way, this is UK. Yesterday, I had the monkeypox vaccination about noon. They tried to give it the new way on my lower arm, but messed up. I guess that's intradermal, right? As you know, this method is tricky, so they have people watching, checking as they train people up. As a result, they gave me a second shot the normal way on my upper arm. Now I have a small sore spot on my upper arm, but I also have a larger sore area and swelling on my lower arm where they messed up originally. I think they came up by that evening, and they are still there on my second evening. Didn't have other symptoms that day, but noon today, so 24 hours after the shots, I had bad nausea for about an hour or so, along with heavy sweating, which continued a bit longer. The nausea was bad enough to do something at the toilet because I recognized I was right on the edge of throwing up. Sadly, here in Santa Clara County, they aren't letting anyone schedule second shots. As far as I remember, I've never had a vaccine response this strong. I think it's a shame they aren't doing antibody tests before and after and checking history for smallpox vaccination, as it would be interesting to see any correlation between people that did or did not have smallpox vaccine. However, I know about the work by A.R., Amy Rosenfeld, which suggests anyway such an antibody test would be inconclusive. Well, maybe for pox viruses it might not be. I just don't know. Anyway, I thought my experience might be interesting, and I wondered if the strong reaction I had might relate to my having had a childhood smallpox vaccination. I don't know if your reaction had anything to do with the vaccine, right? It's hard to know because you can get nausea from a lot of things, but nobody would be able to tell you it has anything to do with your smallpox vaccination or not. I just don't know. But thank you for letting us know. I'm sorry they had to do it twice. Kathy, you're next.
Adam writes, Dear Vincent and colleagues, what a great episode with Michael Merchlinsky. As a pox virologist, I have been listening with great interest to your coverage of monkeypox. This recent episode really brought everything together nicely. I'm writing to bring your attention to a small-scale study I was involved with about 10 years ago when I was a postdoc with Lawrence Eisenlohr. This short report in the Journal of Virology hits on a lot of current arcs on TWiV, including route of immunization with vaccinia, protection against a pathogenic orthopox virus in its natural host, and the importance of both T cells and antibodies. In this study, protection against mousepox challenge correlated better with higher quality CD8 T cell responses than antibody levels. I worked on this study as I was finishing up my postdoc, so I didn't have time to go into detail on some aspects of the story. Hence, that's why it's a short report. Regardless, you may find this paper interesting. Thank you for everything that you do for science advocacy. Cheers, Adam.
So this is a Journal of Virology paper. Epithelial immunization induces polyfunctional CD8 positive T cells and optimal mousepox protection. It's all about CD8 cells in this case. It's interesting. Circulating memory CD8 T cells primed via scarification were functionally superior and conferred enhanced virus control. Control. That's the key word, right? Not protection, but control. Right. Yeah. Oh, that's pretty cool that you did this. This is 2014. Thank you, Adam.
Yes. I met Adam at ASV. He's great.
Yeah?
Yes.
Huh. Didn't meet him. Okay. Who's next? I forgot. Was that you, Kathy, who read that?
I just read.
Yes. Dixon.
John writes, Hello, I am a 72-year-old man vaccinated with 2 doses of the Salk vaccine when it first became available, then vaccinated with the Sabin vaccine when it first became available. These vaccinations were more than 60 years ago. What does the data say about whether I likely am still protected from the disease? Many thanks for considering my question, and many thanks for such an informative podcast. Take it,
So
Vincent.
the problem is that We haven't looked. Look, I'm vaccinated 60 years ago. I think I'm fine, but nobody did a study of 60 years later. Okay.
No, they haven't.
They've looked at 20 years and 30 years, and you still have antibodies. So, Brianne, if you have memory B cells at 20 and 30 years, would you have them at 60 years?
There's some papers that argue about this. I think the answer is less likely for B cells. But I don't know that it's certain. I also think you might have had slightly more boosts than perhaps John might have of exposures.
That's
to
true. I've had
viruses.
a lot of exposures to poliovirus in the lab, right? So John, the recommendation is if you have had a full course, then you're good. I think you've probably been exposed over the years, all right? And you didn't know it because this virus is in wastewater. That means people are shedding it. You may have encountered it somewhere. I think most people have encountered it.
You could encounter one of these people that doesn't have B cells and is a persistent shedder.
It's a spreader. That's right.
So I would say you're okay because I'm not going to get boosted. But if you're worried, if you stay home all the time and you don't go anywhere, you don't encounter people, then you're not going to get infected. But I doubt that you do that. So, but the textbook is the vaccines provide lifelong immunity, right? Lifelong is a squishy word. Depends how long you live.
It's true.
Right?
It's true.
All right. We have two more here. Let's see if we can do these. Brianne.
Bonnie writes, Dear Dr. Racaniello, Dr. Griffin, and everyone else at TWiV. I am a huge fan of TWiV and all that you and all the guests and contributors do for science education and science communication. Thank you for all that you do. I am writing to ask you to say a few words to listeners about the value of a university education and how invaluable the work that universities do is to the scientific community and by extension to humanity. I think this is an important topic to address because in the context of the student loan debate, all I have heard is commentary that focuses on how much student loan forgiveness means to individuals. But no one seems to be recognizing that the whole is greater than the sum of the parts of a university education. First, we all benefit when more people are more educated, whether they finish their degree or not. Education supports us all in learning logic, context, and ethics, no matter what field we choose to study. But additionally, and I know you can speak to this, universities do research that benefits humanity immeasurably. I'm not using the word immeasurably just as a superlative, like, yeah, universities are great, but literally, as in, we literally can't quantify the benefits that universities provide. Even if we think only about the sciences, we know that not only are universities doing critical medical research, but they are also researching topics such as, for example, agriculture, climate change, and coming up with solutions to agricultural problems that are being caused by climate change. Universities study everything from tiny viruses and even the structure of the atom to the vastness of the universe itself. Would we be seeing images from the Webb telescope without universities? I don't think so. Moreover, scientific research also depends on the humanities, which support the sciences by providing training related to logic, communication, ethics, and society itself, to understand the larger significance of scientific research to human society in general. Not to mention, to do the work of finding funding for such research, it's necessary to understand history, politics, and culture. The pandemic in particular has brought science and the humanities together in forming public policy, which is not just based on virology, but on an understanding of how humans in our society behave and on what policymakers think is economically sustainable. When government supports education in any way, it supports science. That's what I'm trying to say. There is a bigger picture to education. Education of anyone benefits everyone, not just the individual learner. And education isn't the only or even the primary role of universities in our society. The generation of new knowledge is even more important than educating students, although educating students is what makes it possible to continue generating knowledge in the long term. Just as vaccination protects others, not just the person being vaccinated, education supports and protects our society, not just the person being educated. I really believe that's true. What say all of you? Bonnie in Milwaukee. And Bonnie gives a full disclosure that says, I have a graduate degree in political science and I'm working on a second one in divinity. I guess I'm a little invested in the university system, or I'm just a crackpot. They're not mutually exclusive. Bonnie, I really like, particularly your comment, education of anyone benefits everyone. I think that's
exactly
a great,
what I was going to say.
That's a great way of phrasing that. I think that in some ways, one thing that you haven't mentioned here is that the importance of the generation of knowledge, but also the importance of teaching our learners how we think about things and how we generate knowledge, as they are not going to just learn the things that are in the textbook today, but they're going to have to deal with new knowledge throughout their lives. So I think all parts of these things are very beneficial. I agree completely.
Dare I say that knowledge goes viral? This is the right show to say it, at least.
I think you could say universities and colleges, right?
Oh, of course.
Yeah.
For sure,
Right.
both.
Yeah.
And, I've had a career, for all the complaining I do, I've had a career at a university and I was allowed to pursue my interests, given an environment in which to do that. And most importantly, colleagues who I could interact with and students to teach, right? It's this unique place that doesn't exist elsewhere. I mean, if you think about corporate research, right? It's a wonderful research environment, but it doesn't have a lot of the other things that a university does. And we train students to go on in all sorts of fields, not just science, but we can get humanities majors and teach them some science, and that will stay with them. And so the university is great because it's all this together. You have a lot of people who are passionate about what they do, and they can pass that on to others. And you're right, university science is terrific. That's where most of the work we talk about gets done, right? The mRNA vaccines couldn't happen without university research. I know a lot of company people don't like to hear that. We developed this vaccine. No, you stand on the shoulders of giants. Come on.
What would you have done if someone at a university had not discovered mRNA?
Exactly.
Bingo.
Right. It all goes down. So I very much have appreciated a career at a university, and I wouldn't have changed it in any other way. I think it's been terrific, and I loved supporting the university, being supported by it, and interacting with all the people, particularly the students that I've interacted with over the years, and giving them inspiration to have incredible careers. It's a really unique environment.
Vincent, you still do that. You still teach your virology course.
Yeah, it's not over. I understand that, but it's an environment that doesn't exist for all the problems that we have complained about over the years. It's really unique and has to be appreciated. And there's in my pick, something related to this comes up, which I will emphasize. And this letter is actually timely with the pick. So, yes, thank you. Go ahead.
No,
Dixon.
I was just going to add that anytime anyone asked me what I did for a living, I said I get paid to think. There is no other way to put that, and nobody else has that privilege, no matter where they are. You can go to work one day and think one thing, and then the next day you come in and you've completely changed course and you're off on another tangent. It's how your brain works. In order to put, we go from A, B, C, D, E in the paper, but we went from A to F and then back to D and then over to Y and then back to it. It's a hodgepodge of data, plural in this case, and you add it all up at the end of your life and you say, what have I done? And you've inspired a whole bunch of other people to do the same thing. And that's what I meant by, and I'm sure you know this too, that's the virology aspect of knowledge.
Yeah.
It can't stop once you start it.
I am pleased to hear, Dixon, that you and I sometimes must describe our work very similarly, because I always tell people that I get paid to spend my time thinking about things that I find interesting and talking to other people who also find them interesting and telling others about them.
Exactly. How unique a life I've had. And I'm sure you're living those lives as well.
It's time for some picks.
Okay.
Dixon, what do you have for us?
Well, we're continuing on the jazz theme, and it will last for some time. I'm either afraid or pleased to tell you, depending on how you're looking at all this. We're into small groups now. We've finished with the big bands, and we're into small groups. And the second most influential small group, as far as I can determine, at least in my opinion, is the Modern Jazz Quartet. It consisted of Connie Kay on drums, Milt Jackson on vibraphone, John Lewis on the piano, and Perthy Heath on the bass. The signature songs are Django, which is a reflection of an era gone by in Paris during the war, the Second World War, that is, and Skating in Central Park. If you want a song which is going to put you in one of the best moods you've ever been, listen to that song, Skating in Central Park. It's romantic, it's thoughtful, it sticks with you after you've heard it. You can hear it echoing in your head, and you get an earworm out of it, but it's a worthwhile earworm in this case. And I've listened to that song in so many situations that if you ever need to relax, just put that one on and just sit back, turn it all the way up, and just turn all the lights out and enjoy it. So the group is a very sophisticated jazz group, and all classically trained. And they came in a very close second to Miles Davis's quintet.
MJQ. Yep.
MJQ.
Well known.
Right.
But they don't tour anymore.
Well, they're not all alive anymore either.
Milt Jackson's dead. Yep.
That's right.
Thank you, Dixon.
You're welcome.
Brianne, what do you have for us?
So I have a New York Times Science article. It's called "To Prevent a Martian Plague, NASA Needs to Build a Very Special Lab." And it is talking about the precautions that are being made and the laboratory space that they are hoping to use for samples that are brought back from Mars. And of course, they start out the article talking about this idea of, well, what if there was a microbe on Mars that was a pathogen to us and we brought it back? But of course, they also have what I've always thought about as perhaps in some ways larger problem of, what if we bring back some Martian rocks and they get contaminated by Earth microbes? How would we be able to decide if were they Martian microbes or were they Earth microbes? And sort of how do we design the place where we are going to study all of these samples when they come back? And so this article talks a little bit about some of the precautions and some of the considerations that these scientists are making for protection of sort of biosecurity on both sides, I guess you could think of, and the ways that they are taking a page from places like the Needle and other infectious disease labs in terms of building those NASA facilities that will eventually study these types of samples.
So Andrea Harrington is the Mars sample curator for NASA. What a cool job.
Yeah.
There are already pieces of Mars on Earth.
Right.
So we're not a stranger to that scenario. But I think we've got enough technology already that we learned with the moon rocks to handle Mars rocks. I don't think they're going to be any different. And I wouldn't be afraid of pathogens, though. Think about it. You need the right host for a pathogen, right?
You do. And I will admit, I've read a couple of science fiction books that have tried to go down this route and have talked about some of the reasons why that would be very hard. So,
yeah, especially if life didn't evolve very far before Mars lost its ability to maintain life.
Yeah.
I once had the privilege of sitting with Baruch Blumberg, who got the Nobel Prize for working out the hepatitis, I guess it's hepatitis B surface protein. But he also became the head of NASA's exobiology group. And we talked for 2 hours, and he was absolutely riveting in terms of his ability to think beyond data, and this is a plural again, of what it would be like to visit places that had life that was quite different from ours. How would you characterize it? And it all can't be based on RNA and DNA. There has to be some other similar but different system that maybe has evolved. We don't know. And I used to read a lot of science fiction as a kid also, so some of them were better than others, but my favorite were the ones that involved the Martian Chronicles by, I think that was Ray Bradbury, if I'm not mistaken. And I particularly liked Ray Bradbury's writing. So what do you think, Brianne?
I agree with you that we probably could use a lot of what we know about bringing back samples from the moon. But given so many of the questions we have about Mars and potential past or current, perhaps microbial inhabitants, I think that thinking about how we would do this is fascinating. I liked the part of the article where they talked about how would we decontaminate something that came from Mars if we don't know if it has similar biological properties. How would we stop them?
Exactly. I off the subject, but related to Mars, I just read somewhere that the scientists at NASA have discovered a way of producing oxygen from the atmosphere of Mars by interrupting the carbon dioxide that seems to be in heavy abundance in Mars and turning it into O2 and water. So,
cool.
We're getting there.
Brianne, when you mentioned how do we know something would get inactivated, I immediately thought about, just think about prions.
Right, exactly.
Yeah. That's right.
But I just, when I read this, it was something I hadn't really thought about, I guess, in a while, or outside of science fiction reading. And it was a nice diversion of something to really ponder.
Yeah.
Yeah, we dealt with prions, right, Kathy? Mm-hmm. We figured it out. We could figure this out.
Yep.
Kathy, what do you have for us?
So I picked an episode by Physics Girl, and I went back and figured out that Rich picked Physics Girl episode 380 in 2016. So if you don't know about her, she's a YouTuber who's got bachelor's and master's degrees in physics. Her name's Diana Cowan. And this one is about spending time in the most remote camp on Earth. And so she goes for 48 hours to the Arctic Circle. She was invited to go there by someone in the U.S. Navy Submarine Readiness Squadron 32. And she mentions that that he attracted her in some ways because a previous one did something about a nuclear-powered subsurfacing. And so she says, I'm going to have to say that for another episode at the end of this one. But she talks about the packing list, the most surprising things, how the place where the bathroom was after she left developed a fissure. And, it's just really kind of interesting, the non-initiated person going to someplace very foreign and it's pretty interesting.
So you asked if you had ever picked this before, but you haven't. But ages ago, Rich picked Physics Girl as a general pick, like episode 30 or something. Mm-hmm. So we can pick it again.
No, I knew he picked it in episode 380. So
380 was it? Okay.
Yeah, 2016.
It's still ages ago.
Yeah. Yes.
But there's nothing wrong with repicking because that means people really like things.
Well, what I couldn't remember was, this is a video that just came out. What I couldn't remember was did I pick it just, several weeks ago? Because I sent it to people, but I didn't pick it for this. So, yeah.
My pick is a little article in Nature, which is open access, so you can see it. It's called Lab Leaders Wrestle with Paucity of Postdocs, or Paucity, by Chris Woolston. And this is, as the title says, apparently it's getting very hard to get postdocs. And they talked to a few people who have had issues. And, it talks about what are some of the reasons. And this is not just in one place. It seems to be in the U.S. and Europe, other places as well. Now, applications are going down. Brexit has caused some issues that contribute to it. Salaries are low. I think also is this idea that, you're doing somebody else's project and you want to do your own thing, and that's, I bet, part of it. But it seems to me a lot of people are going into non-academic careers as well, and they feel that, you know, doing a postdoc is kind of channeling them towards that career. So there was one quote here that was relevant to our one article there, and let me see if I can find it. So they're talking about how people are trying, right here, the dwindling supply of postdocs probably reflects a growing trend of scientists moving away from university-based research. And this person, Lancaster, says it's not just about postdocs. You see really established PIs starting to leave academia. People choose academia for the intellectual freedom, but now there are private institutions offering that same intellectual freedom with better salaries and working conditions. What is academia even offering anymore? And I thought, oh my God, you don't know what it offers if you're asking that question. If it's not just about these intellectual freedom and salaries. I mean, all the things we mentioned is what academia is about.
It's certainly
right?
not about salaries, Vincent. I would take that.
No, I don't think you go to an academic job for salary. You go for intellectual freedom. Okay, I can understand that. And Lancaster says, if we can get it somewhere else, then what am I going to go to academia? Well, all the other things, your colleagues, your students, the ability to influence a new generation of people. All these things, I think, are what academia provides.
The intellectual biodiversity of ideas is the most robust at a university. There is no other place like that, no other institution that encourages that. Well, you know, maybe NIH, but I think maybe national laboratories too. But they're project directed, whereas you and I could have gone in there and talked about basket weaving for a whole year. Maybe we did, actually. I think we actually had a session.
But look, we have 4 people from universities here on this podcast. I never would have been able to do this anywhere else. All right, this is
we feel the same way.
If I were at I don't know, so they're probably talking about institutes that arise, right?
Perhaps.
Where you have intellectual freedom. I couldn't do a podcast at an institute. No, we have an endowment. You got to do research. And who knows how long that endowment's going to last. So, you know, well, I don't think it's fair to say. However, I understand the postdoc issue. So this is a problem. And I think the solution is in part something that John Udell mentioned years ago. We need to depend more on staff scientists, right? More permanent positions of people who can be independent. They don't want their own lab, but they will work within your group. They will have their own projects, their own funding, and they're recognized as a university as a faculty member, right? Which is often a problem for postdocs. And, you know, that's how NIH runs, right? They have a lot of staff scientists and very few elsewhere.
Yeah, I think that's one thing people sometimes underestimate when they think about difficulties of postdocs is that you don't make, you know, the best money ever. We'll put it that way. But you also are expected to kind of move somewhere after grad school with the idea that you are then going to pick up and move again.
Yeah.
You're not necessarily thinking at that point about being somewhere for a particularly long time, at least is the idea with the postdoc traditionally. And that can be a hard sell for people to, you know, imagine that their life is not going to be settled for a while. And I don't think we should underestimate some of that absolutely. And, you know, other types of difficulties for postdocs.
Yes.
All right, we have 2 listener picks. Gary writes, hello, TWiVers. Dixon, in your big band choices, I can't believe you didn't include Stan Kenton. In my opinion, he was the greatest of the later era, 50s and 60s, both in his wonderfully inventive arrangements and his top to bottom superb musicians. For a sample, check out what he and the band do with the music of Bernstein's West Side Story on YouTube. However, as you didn't choose Stan Kenton, I will nominate him as a listener pick. Speaking of superb, I found TWiV early in the pandemic, have never missed an episode since. While there's a lot in the detail of many of your discussions of papers that I don't have the background to understand, I generally follow the thrust of the presentation and discussion, and I find it consistently fascinating. I appreciate the candid but respectful tone almost as much as the content. Thanks for all you do. Gary is in Colorado. Dixon, you have a response?
I do. I wrote, but I will also say that I also love Stan Kenton. He was the first jazz venue to introduce the use of French horns into jazz. I limited myself to only 5 picks from each category, so I'm sorry that I couldn't squeeze in Stan Kenton. Check out Stan Kenton's Peanut Vendor rendition. Amazing trumpet section. I just played it again today. It's as fresh as it was the day I heard it first. And it's amazing, but nobody beats Count Basie, nobody. And then after that, it's a crapshoot as to who else you want to pick, but I think Count Basie was number one. And that's my own opinion. I mean, I'm not saying that that's the truth.
Of course.
This is my own view.
And Miguel writes, in response to some of the comments from episode 929 regarding prevention of outbreaks and efforts being taken, I thought you all might be interested in this project that Microsoft and my organization have been working on in collaboration with others. This could be a listener pick if you would like. And so Miguel sends a link to Microsoft's Premonition project, where they are collecting mosquitoes using these drone delivered contraptions. We had the guys on. Do you remember? A long time ago.
We did. How did that ever turn out?
Well, I don't know. It was interesting, and I guess they're still working on it because the links, you can see they're still doing it. It's great. I had a chat with Lori Garrett this morning, and she said she wants, I don't have a pen here, she wants people to develop a pen-sized device that you dip into water. It pulls up a sample. It does some kind of analysis, and then you plug it into your phone and it transmits it. And she said it has to cost $1. So everybody could participate in environmental sampling. I think that's a good idea. And she said she started talking to a company, but they said, nah, we don't want to do this. It's too bad.
Of course not.
But I think her idea is that you crowdsource surveillance because nobody wants to pay for it on a big scale, right? So you get these little, and then when you get a hit, then you can follow in. So I think that's a lovely idea. All right, that is TWiV 933. You can find the show notes at microbe.tv/TWiV. You can send us your questions and comments to twiv@microbe.tv. If you're liking what we do, consider supporting us. You can give a small amount of money or huge if you'd like. You know, if you have the means, please give us huge amounts of money. Go to microbe.tv/contribute. Dixon Despommier is at trichinella.org, thelivingriver.org. Thank you, Dixon.
You're welcome, Vincent, and everybody else. I look forward to this event. Let's just put it this way. Every Friday I get queued up and I'm ready to go.
Very good. Glad to hear it. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun. If you're going back to find the Physics Girl pick on 380, then you can jump ahead 10 to 390 to hear the show with Building a Better Mousetrap, which is the Premonition project. Yep.
Cool. Brianne Barker's. Sorry, Dixon, what?
I was going to say, is that a comment you would make to a blab mouse? Shut your mouth, trap.
A blab mouse, yeah. Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here. I'll echo Dixon. It's always a great way to end the week each week.
Yeah.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Jolene for the timestamps, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.